Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. I am Waqar Ahmad and this is the second lecture on the chromosome structure. In the first lecture, we have talked about the components which are involved in the composition of chromosome. The chromosome, first of all, we will talk about from the base that the DNA which is present in the human cell. So this DNA, it is all about 6 feet in length. So this is much longer than the microscopic cell itself because this DNA cannot be accommodated in the cell. So there are several levels of compaction that enable the DNA to fit inside the cell. First, the binding of DNA around histones form the small nucleosomes. Next, the tight coiling gathers the DNA with nucleosomes together into higher order structures. The other levels of compaction produce the metaphase chromosomes. Now there are two models for the compaction of chromosomes presented. The two models are the first one is the folded fiber model and the second one is the nucleosome concept. A popular model was the folded fiber model. It was proposed by E.J. Dupra in 1965. The second one is the nucleosome model. It was proposed by R.D. Kornberg in 1974 and confirmed by P. Audet et al. in 1975. The most significant and universally accepted one is the nucleosome model. Now, what was stated in the folded model? In the folded fiber model, it was proposed that the histones were bound on the outside of the DNA coils. So it means that the histone shell around the DNA, that the histones are present outside and the DNA is wrapped inside. This was the folded fiber model. The second one is the nucleosome model. The nucleosome model is proposed the converse. The converse means that the histone particle with DNA around it, that the histone particle are inside and the DNA is wrapped outside. So this is the second model. So the most accepted one is the nucleosome model. A nucleosome, it is the basic repeating unit of eukaryotic chromatin. The nucleosomes are arranged like beads on a string. It looks like beads on a string, or we can say the beads on the necklace. That is, it has the appearance of the necklace. Now each nucleosome is a disc-shaped particle, and the diameter of nucleosome is 11 nanometer, and the height is 5.7 nanometer. The nucleosome containing the two copies of each four nucleosome histones, that is the H2A, H2B, H3 and H4, as we have discussed in the first lecture. So it forms the octamer. The octamer, it is present in the nucleosome. The nucleosome, it contains 146 base pairs of DNA. Each nucleosome bed is separated from next by linker DNA. The two nucleosomes are separated by linker DNA that contain 54 base pair long, and it contains a single H1 histone. The DNA make two complete turns around the histone octamers. Now you will see in the diagram that the DNA, it makes two complete turns around the histone octamers. The two turns, the 200 base pair long, are sealed off by H1 molecule. In a eukaryotic gene of 10,000 nucleotide pairs, it will be associated with 50 nucleosomes. Now each human cell with 6 multiplied by 10 raised to the power 9 DNA nucleotide pairs, it contains 3 multiplied by 10 raised to the power 7 nucleosomes. So these are the calculations and you can find out very easily. Now this is the nucleosome model, this is the octamer, the histone octamer, which is present in the two turns of DNA. This is the one turn and this is the another turn. So the two turns are present here. And this one is the histone H1 protein. This is also the H1 protein. This is the H1 protein. And this is the linker DNA. The linker DNA that are present between the two nucleosomes. This is one nucleosome, this is another nucleosome, and this is the linker DNA. Now there are some non-histone proteins are also associated here. It will be discussed now. Now the solenoid model. The H1 is reported to be phosphorylated just before when the mitosis and meiotic cell division started. So before it, the H1 is reported to be phosphorylated for more compaction of the chromosome. The prophase is the stage during which the chromosome becomes the short and more thicker. The shorter and more thicker, the chromosome becoming the prophase. Now the following types of chromosomal structure can be observed during the cell cycle. The first is the 10 nanometer fiber. First of all, the DNA is 2 nanometer wide, but when it is associated with the histone protein, so first it forms the 10 nanometer fiber. When the nucleosomes are in close opposition, so they form the 10 nanometer filaments, in which packing of DNA is about 5 to 7 times more compaction than the free DNA. So in the 10 nanometer fiber, the DNA compacts 5 to 7 times more from the free DNA. The second is the 30 nanometer fiber. The 30 nanometer fibers, it can be observed in interphase nuclei, and it represents the natural conformation in transcriptionally inactive chromatin. The 30 nanometer fiber, it consists of closely packed nucleosomes. It forms from the folding of the nucleosome chain into a solenoid structure having about 6 nucleosomes per turn. The DNA of 30 nanometer solenoid has a packing that is about 40-fold. Now here the DNA is folded up to 40 times. So this is the 30 nanometer fiber. Now you can see that there are 6 turns of histone. So this is the 10 nanometer, this is the 10 nanometer, and this is the 30 nanometer. So how the 30 nanometer is present? So you can see here that the 6 turns, 1, 2, 3, 4, 5, and 6. So these are the 6 turns of the nucleosome that form the 30 nanometer. Now the radial loop scaffold model. In the radial loop scaffold model, it also defined the further compaction of the chromosome. There are also several non-histone proteins involved in the compaction of chromosome. For example, the topoisomerase 2, it binds to chromatin every 60 to 100 kilobase, and the nucleosome studied 300 nanometer fiber into structural loop. Now there are the non-histone scaffold proteins. These are the non-histone scaffold proteins, these are the non-scaffold proteins, and this is the loop chromatin. Now you can see here that some non-histone scaffold proteins are also involved in the compaction of chromosome. A complex of protein known as condensins. Now some other proteins are also involved in the folding of chromosome, which are known as the condensins. It also acts to further condense the chromosomes for mitosis. Now these and other proteins may gather the loops into daisy-like rosettes and then compress the rosette centers into a compact bundle. Now these are the daisy-like rosettes. These are the daisy-like rosettes, just like a flower you can see, and these are the additional non-histone scaffold components which also help in the compaction of chromosome, and then these daisy-like rosettes, they are compressed further into compact bundle. So this is the metaphase chromosome. Now the complete structure you can see here that first of all, this is the overview that the 2 nanometer DNA wide, it is folded into the nucleosome. This is beads on a string, that it is beads on the string that form a chromatin. So this is 11 nanometer fiber. Then the 11 nanometer fiber further compaction occur, that is the solenoid. This is the 13 in which the 6 turns are present. So this is the 13 nanometer chromatin fiber that form the of packed nucleosome. This is the 13 nanometer fiber. Then further compaction occur that form the 300 nanometer, and then further compaction it formed the 700 nanometer, and then the final mitotic chromosome that we can see we form. So this is the 1400 nanometer, and this chromosome, it contains the centromere and it contains the 2 chromatids. This is the 1 chromatid, this is the another chromatid, and it also contains the telomere regions. So this is all about today's lecture about the chromosome structure. Thanks.